I will be discussing today is leptospirosis. Leptospirosis is a bacterial disease caused by spirochetes of the genus Leptospira. This genus is composed of over 250 pathogenic serotypes or serovars based on differences in the carbohydrate component of the bacterial lipopolysaccharide. It is a zoonotic disease, meaning that it is present in both animals and humans, and is transmitted from an animal host to humans. It is considered an emerging infectious disease in humans, dogs, and cattle. However, it can infect almost all mammalian species. The first case was discovered in 1931 in the Andaman Islands, and it is now considered to be the most widespread zoonotic disease. Although cases do not occur too frequently in the United States, it is estimated an annual 100 to 200 cases occur, with 50% of those occurring in Hawaii. The disease is endemic to many parts of the world, especially those with a tropical and wet climate. Epidemics can and do occur. Usually they are connected to contaminated water sources. They can also occur with changes in human behavior, such as expansion or deforestation, which increases the human exposure to wildlife, or a change in the animal reservoir, for instance, a change in the prevalent serovar. It is possible for an animal to be a host to a specific serovar, show little or no symptoms when infected with that particular serovar. These animals will, however, get sick if infected with a different serovar. As mentioned, leptospirosis is a zoonotic disease that can be transmitted from both wild and domestic animals to humans. Human-to-human transmission is rare, but can occur transplacentally, sexually, or through breast milk. The bacteria is transmitted through infected bodily fluids, primarily urine, and foods that have been contaminated with infected bodily fluids. The bacteria enters through cuts in the skin, mucous membranes, or direct consumption. Once an animal is infected, it can either shed the bacteria continuously or sporadically. Leptospirosis is capable of surviving outside of the host in the soil and water for weeks to months. As was mentioned, leptospirosis is most prevalent in warm, wet climates. The disease is found mainly wherever humans come into contact with the urine of infected animals or a urine-polluted environment. Those that are most at risk of contracting the disease are those that have direct contact with animals, so farmers and veterinarians. Also, anyone who has an increased exposure to soil or water has the potential to get infected if those sources are contaminated with infected animal urine. So campers, miners, sewer workers, and those that engage in water activities are at an increased risk. In humans, the most common signs include fever, chills, headache, muscle ache, vomiting, jaundice, red eye, abdominal pain, diarrhea, and a rash. These symptoms are all common to other diseases, and thus lepto is often misdiagnosed. The incubation period for leptospirosis can range from 1 to 4 weeks, and the disease can last from days to weeks to months if not treated. Disease can present in two forms. The first form is less severe and is characterized by the symptoms priorly listed. The patient might recover for a time before the second stage sets in. If the second stage occurs, it is referred to as Weil's disease. This stage is more severe and causes kidney or liver damage. Leptospirosis is capable of infecting all organs of the body, and if left untreated, can have severe consequences, leading occasionally to death. Case fatality rates in different parts of the world have been reported to range from under 5% to 70%. These figures are not very reliable, as in many areas the occurrence of the disease is not well documented and may not be diagnosed. Initial symptoms can be mild in animals and could lead to a decreased milk production in cattle. In horses, lepto is responsible for 2 to 4% of abortions. Leptospirosis is best confirmed through direct isolation of the bacteria in urine or tissue sample. It can also be diagnosed using dark field microscopy, microscopic agglutination test, and PCR. Prevention and control should be targeted at the infection source, the route of transmission between the infection source and the human host, or infection or disease in the human host. The infection source can be controlled through testing and vaccination of animals. Vaccines exist for cattle, dogs, and pigs, and commercial human vaccines have been produced in France and Cuba. However, these vaccines do not induce long-term protection against infection and do not provide cross-protective immunity against heterogeneous leptospiral serovars. Controlling animal reservoirs through removal of rubbish and stray food from where humans reside can help reduce transmission. Avoidance of areas with potential for contaminated water or soil, proper hygiene, and protective clothing can also help reduce transmission. Treatment of leptospirosis involves antibiotics, typically penicillin or doxycycline. Clinicians should never wait for the results of laboratory tests before starting treatment with antibiotics because serological tests and cultures can take up to several weeks for results to come back. Patients with severe cases also require supportive therapy. Research into leptospirosis is also crucial for identifying the particular serovar that is present in different locations and reservoirs. It is important to understand if predominant serogroups in animals have remained the same or changed over time. This is important to note in order to find out if transmission is occurring from new reservoirs, such as imported animal species. It can also impact the way we construct education, transmission risks, and vaccination strategies. Leptospirosis is gaining approval as a nationally notifiable disease. As such, confirmed and probable cases should be reported to the CDC. This will help keep a surveillance on the spread of the disease.